In the previous section, we have talked about the types of graphs as well as what to take note of when making graphs. In this section, let us look at what to take note of when we are making tables in the paper. First, you have to ask yourself the question, is the table necessary? What kind of information can you get from this table? Is the entire table single or double spaced, including the title, headings, and notes? It should be consistent. If there are other comparable tables presented in the paper or the report, you have to make sure that the format is consistent. Is the title of the table brief and yet explanatory? Does every column have a column heading? Are all the abbreviations, special use of italics, parentheses, dashes, and special symbols explained? They should be explained. Are all probability level values correctly identified, and are the asterisks attached to the appropriate table entries? Is a probability level assigned the same number of asterisks in all the tables in the same document? We'll learn more about what are probability levels and the use of asterisks to indicate the significance later on when we talk about the statistical tests. Are all the notes organized according to the convention of general, specific, and probability? Are all the vertical rules eliminated? Unlike the tables that you see in sample lab data sheets, typically in an official journal paper setting, the vertical lines would not be there. You will only see the horizontal lines in the table. If the table or if the data is from another source, is the source properly cited? The Library Eliza website will show you how to cite a table in the proper format. Lastly, is the table referred to in the text? You always have to make sure that all the figures and tables are being referred to in the text with the appropriate table and figure numbers. Now, when it comes to the captions of tables and figures, there are a few things to take note of. The first sentence functions as the title for the figure or table and should clearly indicate what results are shown in the context of the study questions. The summary statistics that have been plotted, such as mean and standard error mean, should be shown in the captions. The organism studied in the experiment, if it's applicable, should also be included in the caption. The context of the results, that is, the treatment applied or the relationship displayed, etc., should also be mentioned in the caption. Specific explanatory information needed to interpret the results shown in the table, then this would be the frequency done as footnotes and may include a key to any annotation. The culture parameter or conditions, if it's applicable, such as the temperature used, the media that has been used, should be included in the caption when appropriate. Sample size and statistical test summaries should also be included in the captions. Do not simply restate the axis label with a versus written in between and treat that as the caption. This is not acceptable. Your caption should be informative. Now, the acid test to know whether your tables and figures have proper caption is that any table or figure you present must be sufficiently clear, well labeled, and described by its legend to be understood by your intended audience without reading the results section. That means it must be able to be stand-alone and to be interpretable. If your figures and table is too complicated, it may be difficult to understand it in or out of context. Therefore, you should always try to make the tables and figure simple. If you're not sure whether your table or figure meets these criteria, you can give them to another person to take a look and ask them to interpret your results to see if it can be done. This is an example of a table. As you can see, that is the table number with the caption. In this case, this particular bacterial strain is used in this study, and this table shows their characterization. As you can see, the table number as well as the table caption should always be above the table, on top of the table. You can also look at the previous slide to see if this table meets all the criteria. For example, only horizontal lines are seen, the vertical lines are not, and there are appropriate footnotes to indicate that these samples are provided, the source of this sample, where are they from, so on and so forth. This shows an example of a figure. Charts and graphs are considered as figures. As you can see, you have the figure number as well as the figure captions indicated here. And for figure number and figure caption, they should always be below the figure. Whereas for tables, they should be on top. But for figures such as graph, they should be below. In this case, you are looking at the relationship between resistant fecal strain and antibiotic use. That is the title, followed by the caption. The fecal strain were isolated from patients with diarrheal disease caused by bacterial infection. Clearly, the use of antibiotic correlates with the percentage of bacteria resistant to that antibiotic. So it actually explains the trend that is being shown in this particular figure. Note that you start the caption with a label, not a sentence. The title of this figure is actually a label. The sentence is the caption that follows. The label ends with a period or a full stop. Even though it's not a complete sentence, you should also put the full stop here. The figure with its caption is able to stand on its own, as you are able to make sense of this data even without looking into the text. And you can see that this particular graph includes all the elements needed, such as the units as well as the label for the two axes, proper legend being used, and symbols that is clearly explained, and proper scale that is being used. This is another example of graphs that is used in the paper. You notice that under this particular figure, figure 2, there are altogether 2 graphs that were used. You can have more than one graph under one figure, but you will have to label them clearly with A, B, and so on, and the captions for the 2 separate graphs should clearly be indicated. So you can see here A and B. Also take note of the alignment of the figure if you have more than one graph in a single figure. In this case, the x-axis is only shown once because they are actually perfectly aligned with each other. This allows for easier comparison between figures if the scale for the axis is the same. Otherwise, you will not be able to compare between the 2 figures. Now, look at the following examples. What kind of tables or graphs would you use for the following? Number of students in each diploma course in SP, relationship between oil viscosity and frying time, bacterial growth, in this case measured by colony formation, under 2 different temperature conditions over 10 days. demographic data of participants in a sensory evaluation study. Ask yourself this question, and based on what you know from the previous lectures, figure out what kind of table and graphs will be appropriate. After that, look at your own FYP and decide what kind of tables and graphs you will likely be using for which of the variables in your FYP. These will be discussed in class during the classroom session.